Hello, welcome to the new session of Premade Pro. Hope you all are doing well. All the subjects are going smoothly and you all are experience a huge pressure for the upcoming examination. So don't take the pressure, keep your mind cool and just follow the guidelines. In few days, I'll make some short videos or the shorts in which I will try to assess your stress and will try to clear stress busters, and you will definitely get help from those short videos. It will come in a session. Okay, so today we are going to continue with the new chapter that we are ongoing, the molecular basis of inheritance, and we are going to continue with our topic, the transcription, and this is the lecture number 2 of the transcription. And so let's start with the lecture number 2 of the transcription. So write the heading, transcription. Transcription. Yesterday, if you have completed the lecture, you will get to know that I have completed the transcription up to the prokaryotes. So now I will try to elaborately discuss the transcription mechanism in the prokaryotes, in eukaryotes. Okay. So transcription in eukaryotes. Transcription in eukaryote. Eukaryote. Okay. So transcription in eukaryote, RNA polymerase binds to particular sequence in promoter region. Okay, RNA polymerase binds to the sequence. RNA polymerase binds to the particular sequence in the promoter region. Now, what happens? It's known as the Tata box. Tata box. Okay. The Tata box is another name of Tata box is Pribnow box. Now, what happens? Let a gene called a split gene. What is the function of the split gene? Let this is be just the split gene. Let this is be the, this is E, this is I, this is again E, this is again I. Okay. So EI defines E for exons and I for introns. E equals to exons and I equals to introns. Okay. Now DNA is the structural gene. Cistron. Structural gene or cistron. Okay. Now this is the transcription unit. This is the transcription nucleus. Now transcription nucleus contains primary transcript. Primary transcript contains what? Immature mRNA. Immature mRNA, H and heterogeneous RNA, heterogeneous nuclear RNA. Okay, it contains heterogeneous nuclear RNA. Now, addition of addition of 7 methyl guanosine at the 5' end makes the capping. It makes the capping. So addition of the 7 methyl guanosine at the 5' end ultimately makes the capping. So what happens? It's a template independent. Capping is template independent. Capping of the DNA is template independent. Okay. Now, template independent capping also contains enzyme called guanyl transferase. Okay. So capping contains enzyme called the guanyl transferase. Now, again, okay. Now here, this is the 5' end and this is the 3' end. So what happens? This is 7. Prevent the degradation. Here it's prevent the guanyl transferase. What the guanyl transferase does? It prevents the degradation of mRNA from 5' end. Okay. Now the addition of adenylate acid 200 to 300 at 3' end is the tailing polyadenylation. It is poly A polymerase. Okay. Now what happens? Poly A polymerase. Now what happens? It's the 98%. It is 98% intervening sequence. I means intron and E means exon. I have already said intron is the non-coding part. That not form protein. While exon is the coding part and it form protein. Okay, 2%. Now what happens? Okay, what happens now? See, let it see the drawing first. So this was at the first site. This is the 5' end and this is the 3' end. So by splicing means making it in fragments. By splicing, it happens to removal of, this is the process, removal of intron. So intron is the non-coding part. So intron contains nucleus, template, independent. Now the snRNA plus the protein, snRNA plus the protein, snRNA plus protein, snRNA plus the protein. So snRNP, small, that is small nuclear ribonucleoprotein, small nuclear ribonucleoprotein. Now what happens? This small nuclear ribonucleoprotein will make this small nuclear ribonucleoprotein has been done. Okay, now this different color is making. Okay, now small nuclear ribonucleoprotein AAA, that is adenine, at the 3' end contains the intron, that is the non-coding part. Okay, now with the help of the RNA ligase means they will join it. Now, okay, first it will break into fragments. It will be broken into fragments. Now, by the RNA ligase enzyme, what happened? It will be joined as 5' AAA 3', processed mRNA, mature mRNA, processed mRNA and mature mRNA. Okay. Now come to the next part. It is a complex version that I have already explained. Now the simple version. Simple version is DNA transcription. After transcription, immature mRNA, and by the process of capping, these are questions asked in sequence in the NEET exam, which are the sequence of the DNA transcription. A, B, C, D, E are will be given in the options, and the MCQs will be patterned like it. Capping, then tailing, then slicing, and after slicing, the mature mRNA will form, and the mature mRNA will convert into protein, and this process is called the translation. Okay, so this process is called a translation. Now in prokaryotes, what happened in prokaryotes? DNA transcription creates mRNA. It's mature by exonic mRNA. By translation, it's protein. While the protein is made, it can be coupled. The transcription and the translation can be coupled. So can be binded and can be done at once. Okay, now what happens? The introns absent introns absent in prokaryotes. Exons is present, splicing absent, post transcriptional modification, post transcriptional modification is absent, split gene is also absent, okay. So in eukaryotes, three types of RNA polymerase are there. Three types of RNA polymerase is there. So what type? One, RNA polymerase 1. It codes for, this question comes in NEET every year. 5.8 sRNA, 18 sRNA and 28 sRNA. 2, RNA polymerase 2, mRNA is primary transcript and hnRNA. And the third one is RNA polymerase 3, it transcribes for tRNA, snRNA and 5 sRNA comes in NEET every year. This is very much important area. Please try to remember it. Question comes from here every year. Question comes from here, this part, every year. Okay. Question will come from this part every year. Okay. Now, question will come from here. Okay. Now, what happens? So, shall I keep it up to here? Because in the next part, we can start the polycystonic gene. But if I continue with the polycystonic gene, the video will be elongated up to more than 1 hour. Because I am getting a view that people are asking for shorter videos. They are no longer opting for the longer videos. So I am keeping every videos in short, and skipping the short videos will take less time to revise it before the exams. So I am keeping it up here, and I will start from the polycystonic DNA gene from the next, that is the exonic part, from the next video. Okay, till then, good night. Please subscribe this channel, like and share this channel. Thank you.